My job is made much easier than by the preceding talk from Debashish. And since he has already kind of given you an idea of what these small machines are that keep you and me going every day, I will take it on from there. And because I already trusted Debashish that he would give you an excellent exposition, introduction to the subject, I will not be using slides. I will try to convey what we do and the kind of the subject matter on the board here. So as he has told you, somebody at the end of the talk asked the question, how do we think of these things in a cellular context? How do we, most of these machines, in fact all of them are working inside the cells of your body. And so, how big is the cell? How big are these machines? How many of these things are inside the cell? What kind of things are needed to be done by these cells, by these machines at the cellular level? And, how do they do it? So that is the question. Now, as Debashish already told you, at the level of the body and the organ, when we talk about the kind of things that these motors do. Let us talk in terms of forces, for example, because when you think of a machine or a motor, the most obvious thing is how strong is it? What does it, how strong is this machine? So, as I'm moving my hand and, as I'm walking around, that I can pick up kilogram size objects quite easily, tens of kilograms. So the kind of forces that we are used to in our daily life are of the order of newtons, okay? And the distances that we move are of the order of meters, because I'm roughly 2 meters, 1.5 meter tall. However, the kind of relevant length scales and the force kind of regimes of force that we need to know for these machines are much smaller, and they are of the order of piconewton, and 1 piconewton is 10 to the power minus 12 newton, and the dimensions are of the order of nanometers, okay? So Newton meter goes to piconewton nanometer. And so the first question, because the title of my talk is teamwork in molecular motors, so you might ask, what is this teamwork and why is it needed? Now, from most of the talks that you will hear about these motor proteins, it will seem as if that, inside the cell when things work, there is this motor and which is doing this, okay? That is not true in most cases. These motors do not work in isolation. They need to work along with other proteins which help them in some way or the other, and these details I will not go into too much right now. But very often you will see that there are many motors, maybe of the same kind or of different kinds, which are kind of being teamed up at locations inside the cell to do something. So, okay, so that brings me to, so let us say this is the outline of a cell, and here is, let's say, nucleus, and there are many kinds of things inside the cell. Let us not give them names right now, just think of them as small enclosures inside this cell. And so the most important thing to begin with, as we enter the cell, the first thing that you encounter is this membrane. It is a lipid membrane. It is a bilayer lipid membrane corresponding to 2 phospholipid groups which are facing each other like this. If you look at this, if you look at the membrane of the cell with a good microscope, you will often see that this membrane is subject to a lot of fluctuations, okay? There can be invaginations in this membrane. Things are being taken in via this membrane, and something that this cell, in order to live, needs nutrients, okay? And that it gets from the outside, and they are sucked in by the help from these molecular motors. By taking this membrane, suppose something is coming in. Now this membrane will invaginate like that. Ultimately it will break off and will form a small compartment inside the cell, and that has some fate of its own. Now for many of these processes, in some way or the other, you need these molecular machines. And because I have only limited time, I will talk to you about one class of machines, which are the kinesin or the dynein classes of motors, which walk along microtubules inside the cell. So to kind of just give you an idea, now different cells will look very different, okay? So I cannot generalize anything here, but if you take the nucleus of any cell which is in the interface, you will usually have something called a microtubule organizing center here. and from that you will have filaments which go out somewhat like this, and indeed he showed you one image of that. And the ends of these microtubules which are towards the cell periphery are usually called the plus ends, and this is called the minus end. This has nothing to do with the charge of the microtubule. But this plus and minus means that there is an inherent asymmetry along this microtubule. And now this is also part of the cytoskeleton of the cell. For the students, you all know that we have a skeleton, right? I have a skeleton, that's why I'm standing in front of you. Just like that, each of these different kinds of cells has a cytoskeleton which can give these cells different kind of shapes. Now, I don't think you are familiar with the idea of little cars and little things running around your bones, but that's actually what happens inside the cell, that there are little machines which are running around the skeleton of the cell. So it's a dual purpose skeleton, right? It gives you support, it gives you mechanical rigidity, and it also sustains motion of these vesicles, like this guy who had come in, along these filaments back and forth, okay? And that is one of the subjects that we have been very interested in. This is called intracellular transport. Intracellular means transport inside the cell. Now, obviously you may ask, what are the kind of things that are transported? Why is this transport even important? So at a very coarse level, you can immediately imagine that if you left the cell to itself, entropy would take over after some time and everything would get mixed up and you would have a soup. That's not what obviously happens. As Debashish has very nicely told you, you are constantly using energy in the form of ATP, GTP, electrical potentials, whatever, to win against entropy and maintain some order and organization in this cell. And this order and organization is very immediately obvious if you look at any fluorescent image of a cell. There are different things like the mitochondria which will be at specific places, different things will be at different places. Now, in this context, this segregation of different components of the cell at different places, the mitochondria is here. There are endosomes which are there, the nucleus which is here. So all of these things require, at least to some extent, the activity of these motor proteins, which what they are doing is that now, let us say some object comes in from outside. This object that comes in from outside could be nutrition for this cell. It could be even the long list of viruses and bacteria that we heard in the morning from Dr. Kang. Those also do exactly the same thing. They get into these cells and they hijack these motor proteins to go into the cell and replicate their RNA or DNA, whatever it is. So it is very important to understand how these motors they work in the normal state, and when they don't work in the normal state, there are many implications of that, including the most obvious thing is neurological disorders where the neurons, so this is a generic cell that I have drawn for you, and all of you will know that the neurons are very, very long cells, okay? So they can be almost of the order of meters long, and for sustaining things, at the ends of these neurons, which is 1 meter away, you need the proteins that are being made near the cell body, near the nucleus of the neuron, and those proteins need to be transported to the end of the neuron, and synaptic vesicles need to be transported. So all of this is the job of these motor proteins. Now, coming back to this number, piconewton nanometer, why I want to get back to this is typically the things that are moving around inside the cell could vary in size a lot. For example, the nucleus itself could be moved around. One of the situations is where the baby, let's say a newborn baby, when the brain of the baby is developing, the neuronal connections need to be made in the brain. And there is a very well-defined pathway where neurons go from one place to the other, make a connection to the next neuron, etc., etc. And during these processes, if you look under the microscope, you will see that the extremely large forces are required for these neurons along with the nuclei to be transported to different locations in the brain. And for many of these processes, the kind of forces that would be required, you can estimate those things. I will not go into the details of that. They are likely to be hundreds of piconewtons, even up to nanonewtons, okay? So 100 piconewton, or could even be larger than this, okay? Now, so then the question immediately comes is that what is the force that single motors can generate. That is kind of easy to see from here. These motors, the energy source is adenosine triphosphate, and most of the motors that we know till now, there can be exceptions, but more or less all the time, each motor uses one molecule of ATP to generate one stroke, to go through one cycle, okay? And the energy available from one molecule of ATP is 100 piconewton nanometer. You can convert it to your favorite unit, but let us stay with piconewton nanometer. So that is the energy that one molecule of ATP gives you. That is about 25 times thermal energy, okay? Now, this is the energy input, and that is going to be used, and this is force into distance, obviously, and that is therefore the unit of work or energy, whatever you want to call it. So 100 piconewton nanometer, if it is available, the machine which is going to use this ATP is not going to be 100% efficient. Let us assume it's 50% efficient. That's a reasonable number for many of these motors. So you have 50 piconewton nanometer is kind of being expended, is the available energy. And these machines themselves are of the order of 100 nanometers in size, most of them, okay? If they are 100 nanometers in size, the displacements that will happen within the motor cannot be more than 100 nanometer, obviously, and so they are of the order of 10 nanometers, okay? 1 to 10 nanometer. Let's take 10 nanometer as a good number. So then you see that if I say 10 nanometer here, the energy, if you multiply, then you will get 5 piconewtons here, roughly, okay? So typically, most of these machines, I hope this back of the envelope calculation is clear to you, most of these machines will generate forces of the order of piconewtons, 1 to 10 piconewtons. There is some variation, but 1 to 10 piconewtons. Now, therefore, the typical forces required to bend this membrane, make this cell move from somewhere to other, okay? You know, segregate DNA when the cell has to divide. All of these jobs are being done by these same motors, okay? Variants of these motors. So it is kind of clear that a single motor or one or two are not going to be able to generate the kind of forces that are required for many of the cellular processes, and this is obvious. There's no way of getting around it, okay? So therefore, in many situations, these motors need to work in fairly largish teams, and how large is now dependent on the context. It could be 3 or 4 motors, it could be hundreds of motors, for example in your muscles, okay? Debashish showed you that in the thick and thin filaments you have these arrays of motors which are sitting inside your muscle cells, and there are hundreds, thousands, millions of these motors undergo a power stroke so that I can move my hands around. But we are not going to be talking about that situation right now. Let us simplify things a little bit. So in most situations, when you want to understand what these motors do, how do they generate the force, the problem is that let us take cell division. A cell has to divide, so there is DNA in the middle, and that has to be copied, and you have to put half the DNA in the new cell, and this entire machinery has to happen. And for this, what happens is that so you have the DNA here, and then you have you have another pole, and you have microtubules, the same things which I showed you there, stretching around in a certain pattern. And there are motors here, there are motors here, there are motors here, everywhere, which are, doing their job to, separate this DNA here, okay? Now, this kind of a situation these are the same motors which are also doing this transport here, which I told you, okay? But understanding this, there's been a lot of progress and a lot of interest in understanding this, because obviously it is very relevant to cancer, how cells divide and when they don't divide and when they overdivide. What has been difficult to understand from a mechanistic point of view is how do you go from a molecule, which is actually the unit generator of force, to what is happening on the cellular scale. Because these forces couple to each other across largish length scales, across elements which have different mechanical properties, which have different rigidities. So it is not easy to understand what is going on. Now, we need not despair. Of course, work is going on that. What we in our lab have been trying to do is trying to understand this, how teams of motors may be assembled and how they may work inside cells at a slightly reduced level of complexity, and that is at this transport of cargoes inside cells. Okay, what are these cargoes? There are many kinds of cargoes. I will give you some examples. Viruses, bacteria, I already told you. Mitochondria, when neurons are growing and they need to branch, you need energy. And all of you know that ATP is made in mitochondria. So where these neurons are branching, you need mitochondria there for supplying ATP. So these motors will take the mitochondria to that location. For all of these, you need to control the way these motors are working, and it is very likely that not one but multiple motors are driving this motion. So then the obvious, perhaps not so obvious question is that can always motors work in large teams? Now this, if I take one motor, add one more to it, then I would expect that everything would improve. Actually, that is not true. Okay, so there are two classes of motors. There are many others, but to keep the discussion simple, and here I will rub out a few things. I do want you to remember this. So suppose this is an object, it could be a mitochondria, it could be whatever, it has to be moved inside the cell. So there are motors which, now again there are exceptions to everything, but just I'll generalize a little bit. There are motors called kinesins, I will call them K, okay, which are attached to this guy, and they generate force in this direction towards the plus end of the microtubule. Kinesin comes from the word K-I-N-E, kine means to move, okay? And there are dynein motors, dynein, D-Y-N-E-I-N, this is derived from the word dyne, which is a unit of force, again, which are also attached to this, this object, which generate force in the opposite direction, okay? And there has been a lot of interest in understanding how the opposing activity of these two classes of motors finally decides where this object is going to go, okay? Because that, to a large extent, is responsible for localizing things at different places inside the cell. Now, let us say this is a virus, okay? Instead of a virus, you could have a mitochondria, you could have lipid particles, many different kinds of cargoes. And what is happening on a virus in terms of how kinesins and dyneins work may not be what is happening on a mitochondria or on some other organelle, okay? So there is no general answer to these things. And so in this context, when we thought of looking at this, we decided to focus on two kinds of cargoes. Now I hope what you understand what I mean by cargo. If you think of this object which is moving inside the cell, the typical velocities are microns per second, okay? Now, a typical cell will be 10 or 20 microns across. So for the cell, that is a fairly high velocity, okay? So you'll get across the cell in some 10 seconds or so. Now, when something is moving inside the cell, we call it a cargo, and there are ways to find out that this is driven by the motors or it is diffusing, but let's not get into that right now. The cargoes themselves are typically of the size of a few microns, okay? And I told you that these motors are of the size of 100 nanometers, which are 1/10 of a micron. So you could have more than one motor. There is enough space for these more than one motor to attach to this cargo. So we decided to focus on two cargoes which are carried by motors, and I will, in the remaining time that I have, I'll focus on two cargoes. One is called phagosome, and the two is called lipid droplet. I'll briefly tell you why we work on these. And so both of these, obviously, because I'm talking to you here, are cargoes driven by these motors. The phagosome is a generic term for any solid object that is ingested by your immune cells, okay? Your immune cells are your first line of defense against all kinds of pathogens which, which have the possibility of infecting you. For example, a phagosome could contain Mycobacterium tuberculosis bacteria which are ingested. And normally, if it is, if it is a benign bacteria, your immune system is supposed to kill these, okay? And however, Smart bacteria or viruses like mycobacterium, one example, they have found out ways to avoid your immune system. And one of the things that these bacteria do is that they subvert this motion and they adapt it to their own advantage, and that is how they survive inside cells. What is this motion of phagosome supposed to do? When a phagosome, when a mycobacterium or whatever else is ingested from outside, it is supposed to be ultimately taken to an acidic organelle near the nucleus of the cell. That acidic organelle is called the lysosome, where this phagosome, generally, if everything goes right, it fuses with the lysosome, and in that acidic organelle there are enzymes which can kill this pathogen. But that doesn't happen for pathogenic bacteria. So what we were able to show a little while earlier is that on these phagosomes, the dynein motors, which are the motors which will take you towards the center, okay, towards that nasty lysosome where you are supposed to be killed, those dynein motors are present in fairly large numbers. And before that, my work in my postdoctoral tenure, what I was able to show that each of these dynein motors has a unique ability to engage an automatic gear-like mechanism, which is present in its inner machines, which I will not be able to go into right now. What do I mean by that is that all of you, if you drive a car or a scooter, you know that you want to go to uphill, you shift to lower gears, right? And that is when you can generate larger force. You change the engine to wheel coupling ratio using your clutch. And when you shift to lower gears, then you can generate more force, but you go slowly, okay? So something like that the dynein motor seems to be able to do, that when you pull it back, now who's pulling it back? Probably this kinesin motor, who is generating force in the opposite direction. The dynein motor seems to slow down rapidly and correspondingly generate higher force. So the work done per unit cycle does not change. Now, so then you may ask, okay, how does this help? Okay, here is a dynein motor which can shift gears, so why should I care? What we argued in a more recent paper is that this ability to shift gears in response to opposing load may allow many of these dyneins to work together as a team, okay? How does that work? So imagine that I'm here, okay, and I'm pulling something, I'm pulling a spring from here, and there are 3 more dynein motors behind me, and we all start to move forward. You can intuitively see that the first motor, which is me, first dynein will see the highest opposing load, okay? And if that happens, if I can rapidly slow down, whereas the ones behind me are not slowing down, they are going fast because they don't see so much load, they are in 4th gear, I am in 1st gear, then there will be a tendency for us to bunch together. As we are moving along these microtubules, we move with differential velocity depending on the load, and this allows these dyneins to bunch together tightly, and that is how probably based on experimental evidence, they are able to share mechanical load better because spatially they are close together to each other. Subsequently, what we showed in more recently is that these dyneins and kinesins actually do engage in a physical tug of war on individual phagosomes and endosomes on cargos, and the mechanics of that, there are special adaptations in different motors. There is a catch bound like behavior where you attach tightly to something when you are being pulled back, but I won't have the time to go into those things. So what we were more recently we have been very interested in is the lipid membrane. Just like I told you that this cell has a lipid membrane outside it, each of these cargos, the phagosome, endosome, mitochondria, everything needs to have a lipid membrane, otherwise it will mix up with everything else, right? So the lipid membrane of the phagosome, what we found is that it is very dynamic, okay? As the phagosome comes inside and as it matures during its transport, there is a significant increase in the amount of cholesterol on this membrane. And this cholesterol, what it does is that it leads to, in cohort with some other features, leads to segregation. It forms to formation of lipid raft-like domains on the membrane of the phagosome, and these dynein motors seem to cluster into these lipid rafts. What is the advantage of this? You can see that if you have a spherical cargo, which if it is large and it has to move on a straight line, which is the microtubule, you have a contact area problem, right? If you just dump motors, they are not all going to be able to reach the, if you dump motors on this cargo randomly, they are not all going to be able to reach the microtubule. But if you cluster them into tight pockets, there is a high chance that this, if the right cluster is at the right orientation, they will be able to reach this microtubule, and that's what we were able to show. And indeed, we were able to show that certain pathogens like Leishmania are able to disrupt this cholesterol-dependent clustering of dynein motors, and that may be one way in which Leishmania avoids being taken here, okay? The other thing that I'll just take 2 more minutes, that we have been very interested in is completely different from this, and you may wonder what is there to do with motors here. We have been very interested in understanding how the liver, your liver, establishes lipid homeostasis in your body, okay? Now, where does the motor come into this, obviously? The liver is made of hepatocytes, obviously these are cells in your liver, and one of the major jobs of the liver is to secrete a molecule called triglyceride. Okay, this is an energy-rich molecule, and this energy-rich molecule is secreted from the liver in the form of a particle called very low density lipoprotein, VLDL particle. All of us see VLDL in our medical reports at some age, and we start getting concerned about VLDL. So the liver secretes VLDL at a certain rate. Now, the problem is that this VLDL, which the liver is secreting into your circulation, is derived from particles called lipid droplets, which are sitting inside the hepatocytes in the liver, okay? Liver can rapidly catabolize these lipid droplets, okay, and package them into VLDL part. I'm simplifying things here, it's not this, but it rapidly catabolizes the lipid droplets, chews them up, packages them into VLDL particles, and send them out at a constant rate so that the energy supply is maintained in your body. But then there is a problem, okay? And the problem is the following, that when you go to bed every night, when you fast, your brain sees that, okay, after some time the glucose levels in your body are declining, and you cannot use glucose as an energy source. You need a different energy source, and that different energy source is this triglyceride, okay, which is packaged into these lipid droplets in many kinds of cells in your body, and the one major place where these lipid droplets are sitting is in the adipose tissue. I'm not a good example of showing adipose tissue, but in your adipose tissue. Now, so the brain sends a signal to your adipose tissue, and the lipids there are chewed up and they are sent to your liver first, okay? Because the liver is the postman who's going to send out everything. Now the liver, overnight, you can see this if you take a section of the liver, the amount of triglyceride, lipid in your liver is about 5 to 6 times more than when you went to bed, okay? Now I told you in the beginning that the liver rapidly catabolizes the lipid droplet and send them out as VLDL. If when you get up in the morning, your liver is doing the same thing, then you are in deep trouble, okay? Because if with such a huge amount of lipid droplets sitting in your liver, if you continue to do the same thing, your VLDL secretion would go up 5 times, okay? And that would saturate all your other organs, your heart, or the places which don't like lipid, and that would be very bad for you, okay? The liver can prevent this somehow, okay? And what we have found out, we are trying to go through this, is that the kinesin motor inside the hepatocytes of your body is controlled by insulin, okay? And it controlled by insulin, and depending on whether you have insulin or not, these lipid droplets are actually taken to locations inside hepatocytes, which is actually the smooth endoplasmic reticulum, where this lipid can be chewed up and sent out as VLDL. I think I will stop here. I did not talk so much about teamwork, but I guess I apologize to you, Devashish, for that. Thank you very much.
Why do we need the kinesin to pull back against the dynein, and what other functions does the kinesin have other than that?
No, so this is one situation where I talked about intracellular transport. Kinesin does many other things in many other contexts, in cell division also, but basically it's a force generating motor. Now here, what I told you, this cargo, let's say the mitochondria, has both kinesin and dynein. Unless you have both kinds of motors, how are you going to locate this mitochondria at different places? If you had only dynein, you could go only one way, right? So you need to control the opposing activity of these two motors, one which is going to take you this way, one is going to that way, to kind of localize themselves spatially in a defined pattern, and this pattern need not be constant, it can change.
Hi.
Can you ask that question later? Maybe we are running. Actually.
So when you were talking about differential velocities of dynein along the track,
Right.
you were talking about the leading dynein being,
No, it's this one, yeah.
you were saying that the leading dynein was being retracted
slowed
by
down,
some
slowed
spring-like
down more.
mechanism. What is that spring-like mechanism?
It's not a spring-like mechanism. The spring is when I said spring, I meant that you're trying to pull a spring like this, okay? That was the load against which you are working, but that is not the mechanism, that is just a general term for load, okay? Now when you apply load, there must be some mechanism within the dynein motor, within the molecule, which allows it to slow down rapidly, quote unquote shift gears.
But if you have 3 dynein molecules along a track, why should the ones behind
No,
be
because they are all
experiencing less load?
No, because they are all attached to the cargo. The dyneins are not directly feeling the load. The load is being felt by the cargo. Okay, so the dyneins are individually not feeling the load. The dyneins are feeling the load via their mechanical connection to the cargo, and it is the cargo which is being pulled back. So in that situation, the one which is at the front is stretching the most. The other ones are kind of floppy and kind of tagging along at the back.
Worked out how these triglycerides, they are actually carried by these proteins. Proteins.
So they're packaged into lipid droplets, and the lipid droplets are moved in hepatocytes and many other kinds.
No, these are triglycerides.
Triglycerides.
Totally hydrophobic.
Esterified molecules.
Esterified,
Right.
totally hydrophobic.
Right.
And proteins are hydrophilic, and how they really attach.
No, so that's what I'm saying. So what happens is there's a particle called a lipid droplet, which is a micron-sized particle. Inside that the lipid droplet has a phospholipid monolayer membrane. Okay, and inside the lipid droplet there are these neutral particles like triglyceride, esterified cholesterol, and many other particles which are packaged inside. But outside you have a monolayer of phospholipids, so that the head group of the phospholipid is facing outside.
It is carrying entire thing.
Yes, and this motor is attached to this monolayer. Yeah.
How are these?
I could have shown you these movies, but sorry.
Yeah, how are these kinins and dynesins? How are they arranged? Is there some scheme? Definite scheme of their arrangement?
Arranged on what length scales?
Like, they have to pull the cargo.
Right.
So I mean, is there some track or anything throughout?
No, so the track is the one on which they are walking.
Yeah.
And while they are walking, they are carrying this cargo. Do you want to ask how they are arranged on the track or on the cargo?
Yeah, how are they arranged on the track? Yeah.
On the track.
Yeah.
When you talk of how they are arranged on the track, most of the kinesins and dyneins are, if they are not attached to a cargo, then they are inactivated.
Okay.
Okay, you don't want to be using up ATP all the time. There are mechanisms by which they are inactivated, but I will not get into that. But they don't just go and sit on the track doing nothing.